It was on the news. I'm Allan Gilbert, and I'm sitting here with Max, not behind the glass, and Scott J. Carroll. How you guys doing today?
Pretty good.
Yeah, I'm doing good.
Good.
I'm not behind the glass for now.
You're not. We're not. I like letting you loose in the studio. It's a lot more fun that way.
Probably not for the best. I was going to say probably for the best, but I don't know about that.
I don't know, man. Last time I was here, you were out of the glass, and it was a great time.
So, well, guys, guess what? We're going to talk about things that were in the news.
Okay. Cool.
One of the things that keeps happening is a lot of news programs are dependent on earlier news programs. So you see something in the news and you'll see a little tidbit, and I'll throw in some of my favorite news articles are, things in outer space or some new biological, some new fancy insert here that you get to see about in the news, but you don't always necessarily figure out what dominoes fell to get you to that point.
And that's fair.
So I thought today what we would do is we would look at CRISPR.
Okay. Yeah.
And explain exactly what that is and basically why we should care.
And that it's not something to do with food.
Mostly. I can tie anything back into food.
Man, I was sitting on the joke about the drawer in the fridge.
Yeah.
But
well, we'll get there. I promise you. All right, so let's talk about CRISPR. What the heck is CRISPR? CRISPR is nothing more than our favorite two words, genetic engineering.
Okay, I completely understand, and I'm good to go.
All right, well, and how many great horror movies, science fictions, and just in general public menace things have begun with the words genetic engineering.
Mm.
All right, well, the first thing I'm going to point out is corn. Yeah, corn's a normal everyday plant, right?
Well, yeah.
No, it's not.
But no.
Corn does not exist. Corn does not exist in the wild. There is no wild corn out there in a field somewhere waiting to be discovered. Corn is utterly and completely a man-made plant.
Go on.
So for however long it took to create corn, the indigenous people of the areas, especially we're gonna pick on Central and South America, had a type of grass that made seeds that were edible, and through a long process of crossing this one with that one and that one with this one and seeing what you got, eventually they developed what we now see as corn.
So they were Gregor Mendel before Gregor Mendel.
Long time before Gregor Mendel. Now, since you brought up Mr. Mendel, why don't you explain Mr. Mendel?
Gregor Mendel is kind of the father of... Remember you would do Punnett squares in biology?
Right.
Where you would have, you do it's not tic-tac-toe, but you set up the tic-tac-toe board and you see which genes are dominant, which genes are recessive.
Right.
He figured that out by crossbreeding plants.
Yes, peas.
Yeah. And he would look at which ones flowered more, which ones produced more, which ones had a different color. And through crossbreeding, he was able to determine, if I've got two yellow flowers, they're going to make a yellow. But if this yellow flower came from a red flower, then there is a chance that its offspring could be a yellow or a red.
Correct.
And those... So he's the father of genetic sciences, basically.
Mm-hmm. We blame all this on him.
Yes.
However, with or without him, this is how we have been developing all of our domestic animals and pretty much all of our foodstuffs. Because if, you happen to like potatoes, well, you happen to have this really, really cool looking potato that's kind of small and golden. Well, it makes eyes. So basically it clones itself. Mm-hmm. And potatoes also flower. And we'll pick on Mr. Luther Burbank here for a moment, who got into also producing plants the old-fashioned way, crossbreeding them. And he was looking for a particular color, size, productivity, things and this is why there's still a wonderful Burbank seed company out there bearing his name, because this was how genetic modification has always been done. So now, and I'm going to back up to one of your favorite movies.
Sure.
And in that favorite movie, there is a animated display of Mr. DNA.
Dino DNA.
And basically, they made it sound like we could just take a bunch of DNA, and if there were holes in it, we could fill it in and make something.
Yeah, that's what they did.
That was the premise of a fictional story, I may throw in. And when we come back, we're going to make it not so fictional.
Ooh.
It was in the news. I'm Allan, and I'm sitting here with Scott and Max, and we are kind of discussing genetic modifications.
Just casually.
Casually, it can
that's
happen.
something everybody talks about nowadays.
Well, in a lot of ways, you're not wrong. They may not know that's what they're talking about, but we see a lot in the news about farming, weed control, chemicals, the pests, the Olympics are going on at this particular time. Well, the Olympics back one, we were really worried about disease borne by mosquitoes, which caused human deformations. And so we do talk about genes, gene manipulation, and gene editing a lot. We just may not quite realize that's what we're talking about.
Everybody's
kind of staring at me. It's kind of like, yeah, okay. Well, this is why we're kind of like, it was in the news. But was it hugely in the news? Was it drastically pointed out that this was that? Or is it just a related topic? Well, everything we're talking about these days, especially when it comes down to food, pest, disease, health, we read wrapping up a pandemic. Congratulations to a virus looking at humans going, you're now tasty. And I've figured out how to make a protein to let me jump out of whatever animal I've been living in and come live in you guys. And this is not a static process. This is not something that happens once and then, it never happens again. As a matter of fact, the CDC is watching, I think at last count, was well over 100 different viruses that could very possibly at some point in the future figure out humans.
We like to think we're complicated. We're not that complicated.
Sometimes we are, sometimes we're not. So kind of welcome to this very muddy water that we look at as genetic manipulation or genetic engineering. Okay, a case in point for genetic engineering, we now have light up, glow in the dark aquarium fish.
Yeah.
Okay. We now have the ability to clone something.
Mm-hmm. They tried to make a cat that would glow if it was exposed to radiation.
Oh, that was interesting. There is a whole company in Korea that, if your little Fifi was an exceptional example of, that particular breed, and Fifi is about to expire, you can have Fifi replicated. It's okay, so this is going on. With this, we're also getting into species preservation. We're looking into food. We're looking into crop control, pest control. And let's face it, how many of you would actually miss mosquitoes?
It depends
no.
upon whether or not we would miss them. But my major question is do we need, if we eradicate a whole species, they might have actually been like a keystone species. Like, even if we don't necessarily like the hornet, for example, if we up and destroy the entire hornet population, for all we know, we might be kind of like really messing up the ecosystem here.
Well, and that is where we're going to get into the words unintended consequences.
Science is other side.
Yeah, well, welcome to everything we do and everything we don't do. We love bananas. We love bananas. We love bananas. But because we love bananas so much, there's very little diversity in the actual banana population. That's kind of a shocking thing for people to kind of go, you do understand that if one plant disease comes along that finds the I think it's the Cavendish banana that we find so tasty, that's a very delicate population of, case in point, in the potato famine in Ireland. One disease got imported and suddenly nobody had any immunity to it, and an entire crop failed.
Mm-hmm.
So everybody kind of goes, well, what does the potato famine have to do with genetic engineering? Well, potatoes are not native to Ireland. Potatoes came from the Americas. So, it had to come from somewhere. So we brought that in, but when you bring something in, you bring in it, and occasionally you bring in its critters.
Yeah. The other, like Max was saying, the ecological implications of it. If this is a plant, someone goes and sees a plant that's really, really pretty, and they like it, and then they decide to bring it over to, let's say, America, let's say, I don't know, Atlanta.
Right.
And they plant it, and then all of a sudden the entire city is covered in kudzu.
Oh, you mean like the World's Fair of 1894 where the Japanese exhibit was giving away samples of it?
Yes, exactly like that one.
Or that the agriculture department later went, this thing would be really great for preserving hills. Well, all right. Well, ladies and gentlemen, welcome to the sticky wicket, if you will, the web of this. And we haven't even got into the genes yet. Because if you go down the Tennessee River and you really gun that boat engine, there's a pretty good chance an Asian carp weighing about 30 pounds is going to airborne into your vehicle.
Oh, free fish.
Yeah, well, and that brings up a related topic called eDNA. So DNA is going to be a bigger and more widespread topic than people really honestly get. Max, you and I talked recently about atmospheric eDNA.
Exactly. You just swab a little bit of the air and let's see who's been around this little environment.
Exactly. And they were testing it at zoos. And the funny thing is they were finding, the meerkats are here at the zoo, but we were finding the meerkat DNA in the sniffer 250 yards away from the meerkats. So apparently the atmospheric eDNA worked, and we were able to pick up and find that these animals do live here. So with all of this said, DNA is really, really a hot topic, and it's such a hot topic that you can now basically buy in bulk the amino acids that make up DNA.
Yeah, we talk a lot about DNA, and a lot of people will talk about it, and then they won't really, they can't pinpoint what makes it up.
Yeah.
What DNA, and it's just 4 amino acids.
That's it. We come down to 4 building blocks. And if you remember your Jurassic Park, they had built in the lysine, the lysine
contingency.
That if we quit feeding the animals lysine, then of course the entire system would break down and the dinosaurs would pass away. And I believe it was the chaos theorist who kept saying life will find a way. And the herbivores started eating things with lysine in it.
Mm-hmm.
And of course, the carnivores were eating the herbivores and thus getting their lysine.
And then you had the other side of the science was they had site B where they didn't have the lysine contingency at all.
Right, at all. So welcome to this kind of thing. Let's talk about that gene editing we saw in the movies. Had the little animated guy comes dancing out and they're trying to show you the gene stream. And then he's kind of like, and then we just matched it up with a frog. And of course, it didn't really work like that. It does, but it doesn't. And especially at the time that movie came out, he was showing you probably 100 years of gene splicing to get those kind of results.
Because Jurassic Park came out in 94, I
The
believe.
book several years earlier.
Yeah, the book
Right.
several years, and when Michael Crichton wrote it, it was, here's what we know, here's what we could probably do. But even in 94, the genome sequencing was not at that level
No.
yet.
Now let's fast forward to 2020, 2021, 2022, where DNA sequencing is not just a science, it is a big business. Well, it's where we just got our vaccines. So in a very short period of time, a lot of science happened, and DNA and rDNA, which is basically think of your RNA as the guys who do what the DNA says do. It's kind of the easiest way to, I'm not going to get into an in-depth scientific explanation of nucleic acids, but DNA is basically the boss. RNA is basically who does what the boss tells it to.
And the tRNA does what the RNA tells it to do.
You know,
It goes for
we can keep going into that, but basically the idea is that DNA is a programmed sequence that can be replicated. Watch any CSI show, any show with a criminal investigation department, and you will hear the words, let's get this to the DNA department. And I'm sure that every law enforcement officer on the planet cringes when they hear that.
It doesn't work like it does on TV.
It doesn't work anywhere near as fast, but genetically analyzing things is a very much a reality and a huge business today. So as we wander deeper into the gene pool here, when we come back, we're actually going to get introduced to a bacteria, basically the Cas9, which in an odd sort of way has truly accelerated the science. It was on the news. I'm Allan Gilbert, and I'm here with Max and Scott, and we are wandering through the wonderful world of gene editing, and we're going to talk about Cas9. Cas9. It's a bacteria, better known as Crispr-Cas9, and, well, this little guy has, I guess, made gene editing a whole lot easier.
Yeah. It's able to help pinpoint where on the gene you actually can edit.
So basically, as we get genomes programmed into the computer, you can kind of pinpoint the spot, and for lack of a better word, you can cut it at this location and replace, remove, or insert something new.
It's basically saying here, right here, because the Cas9 goes in with something called a PAM marker. And that's where it says on the DNA, okay, right here, here's the X. That marks the spot. Go in and cut. And then you're able to input the corrected or the repaired genetic sequence for that small bit.
Now, I'm going to point out that genome editing has been around since the 80s. This is not new. So, for those of you listening right now and going, oh, No, we've been playing with genes, and I'm going to go back to crossbreeding and crop development. We've been playing with genetic development for thousands of years. So this is nothing shockingly new, but it's kind of like getting a smartphone. You know, in my lifetime, we used to have rotary dial-ups. I'm looking at Max and go, you have no idea what I'm talking about.
Hey, I do.
Oh, that's right. He's seen an old movie.
I've seen stuff. It doesn't mean I've experienced it.
You didn't. You never lived until you had to stick your finger in this thing and slide the thing right in and went tick 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 tick.
I know the dated references from Seinfeld where you had to like go into a phone booth to call people or carry around a brick.
Yeah, you watch it. You watch a movie like War Games, and he's having to hack a phone booth. Tick 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 tick. All right, guys, technology and everything advances dramatically. So now with the genetic engineering capability of a Cas9, this gives us that exact targeted spot. If we know what we're looking for, this can let us find it and hit it. So before we start screaming gremlins we're going to go back to Jennifer Doudna and Emmanuel Charpentier, and they published their findings back in 2012. And they were using programmed RNA to edit the genomic DNA. And, okay, we're here. It exists. The technology's been tested. It's been peer-reviewed. And it is now actually more or less in production. As everybody looks in stunned silence. That's great for radio, guys. So we're here. We're here. And now, Scott, you were just talking about a game on your PS3.
Well, when I first got the PS3.
Right.
Because we're a couple generations after that. When I first got the PS3, it came with this thing called Folding at Home. And I didn't know what it was at first. And then I opened it up and looked it up. What they ended up doing, this company discovered that the processing power of the PS3 was better than the computers they had in their science labs. And they realized, well, if we network the PS3s together when they're idle, when people are sleeping or they're not using them, we can run our program. And what their program did is it simulated how proteins unfold to replicate and then refold. And in that folding process, sometimes things go wrong. And that's when we'll get little genetic errors in the replication process.
Right.
And they wanted to know what happened. So they were running simulation after simulation. When they hooked it up to the PS3 networks, all of a sudden they had a worldwide access to these insane processing speeds that they didn't have access to before, and their information flew in. And they were able to make advancements because of that. Because everybody who had, they just discovered, oh, this gaming console is way better than our science computers made for science. And people just went, wow, you know what? That's cool. I'll leave my game station on overnight while I sleep.
Okay.
And science happens while I'm sleeping.
Okay.
And they were able to get so much information that now companies like AMD and Sony, people that make graphics cards and make these processors, are working with them directly saying, yeah, we can do this. And they've been able to put a bunch towards COVID research, towards other protein folding research, and it advanced that field of study dramatically just by people going, you know what, I'll just leave this little thing on. Because the cool thing was, when you ran the program, you got to watch the simulation. So you could watch this massive protein string wiggling and popping open and popping closed. And for the most part, I had no idea what I was looking at. I was going, well, that's just really cool. That's science happening right there.
See, parents, video games are saving people's lives.
It's true.
Well, at least the leaving it on and not playing it. But
no, don't add the qualifier.
Don't add the qualifier. Just say, play with your PS. And what are they now? 5, 6, 7, 12. It's
5 at time of recording.
Yeah, oh my gosh, it's just amazing. So here we go. So we're here. This isn't theoretical. This isn't, this is going to happen sometime in the future. There are people really honestly looking at this going, so what could we do? Well, the Zika scare in Brazil brought up a huge thing about, pest control. Well, all right, we don't want to wipe out every mosquito on Earth, but we would like to at least put a big dent in the one that carries Zika. So one of the proposed things that could be done would be to basically program a mosquito to go out and do little mosquito things and procreate into little mosquitoes, and the new little mosquitoes would all die before they grew up.
A programmed-in lysine contingency?
Basically a poison pill, if you will. So you would grow little boy mosquitoes that don't bite people, only the girls. So you'd grow lots and lots of little boys. You turn them loose to go make lots of little babies that will never grow up. So I'm just throwing out there. I mean, this is a real consideration that somebody has put out on the table. And you kids now begin to back up Mack trucks full of questions. Does it work? Does it not work? How well does it work? There's a lot of testing to be done. But, just in the kind of the thought that a lot of just human inconvenience and suffering could be avoided by making this one tiny little change in just this one species.
I think there's a lot of the overall fear of, like with all science, with all new science, it's fear of the unknown. It's what's going to happen if we do that. Are we going to accidentally create a super mosquito? Or are we going to inadvertently eradicate them all? And then it kind of veers into slippery slope arguments.
Well, then we can get into talking about, instead of eradicating a species, when we come back, we'll talk about what about bringing something back? Well, there's the Tasmanian tiger, the thylacine, that is gone because of human interaction.
We talked about the woolly mammoth a while back.
Well, we're going to bring up that old woolly boy again, and we're going to bring up the dodo and several other species, and the fact that there is even now a genetic arc project in action right now to store genetic variations just in case we do lose some more species.
It's like the Svalbard seed vault.
Exactly. The seed vault is another example of, again, genetic storage.
Perhaps you could explain that a little bit to the listeners that don't know about old Svali.
Oh, well,
it's wonderful.
We'll dive into that as soon as we come back. It was on the news. I'm Allan Gilbert, and I'm sitting here with Scott J. Carroll and our infamous Max out loose from behind the glass. And we're having a little fun talking about genetic modification, manipulation, CRISPR.
It's all the fun stuff.
It's the fun stuff. Well, the thing is, this is, in my lifetime, this has gone from, honestly, science fiction to, wow, we can use this in court, to, wow, makes a great movie. At this point, I'm going, and at dinner tonight, man, that was tasty. Just to kind of put things in perspective, this is one of those weird topics a lot of people know very little about. There's a lot of misinformation, and even if you had all of the information, you still look at it and go, yeah, let me think about that for a minute.
There's some ethical questions.
There are a lot of questions. However, I'm going to point out, all we are doing is following the technology. We, as modern humans, have been doing this since we decided we liked, cotton. or sheep, or we decided that we liked chickens, or we have been manipulating the genetic outcome of the world around us for thousands of years. If you want a great example of that, just look at your dog registry.
This is the purebred. This is the
Well, these are the standards that make this a breed of dog. Well,
We can't really say purebred anymore because they're about as human bred as you can get.
Well, it's outside of a wolf, or a dog-like animal, every species that lives with us is kind of been manipulated, one level or another. So, now Max, you brought up the global seed vault.
Yes, old Svalbard.
All Svalbard. Well, let's go to Norway.
Oh, yes, please.
Let's go to
Just
Norway.
after we got over ice apocalypse, you're bringing us to the cold again.
And we're going to go to Spitsbergen in the remote Arctic. It is up there in an archipelago, and the seed vault is designed to store duplications of all kinds of seeds from around the world. And this is basically a backup, if you want, literally a genetic backup program of everything we use for food.
Mm-hmm.
So, if there's a particular kind of chickpea that only grows in a country, chances are Svalbard has a sample, a frozen sample up there in the Arctic, so that we have safeguarded this particular genetic variety.
Did they choose this particular area in Norway both because it's isolated and because it's cold? Because it pretty much makes like as big as a refrigerator as you can.
And you are exactly correct.
If we only had a ding, 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 ding sound effect.
I say you need a ding. You need a winter bell. So what a great idea! And guys, this is all about genetics. Again, I pick on the banana. The banana is a vulnerable species, and it is a huge money crop around the world.
Oh,
It is vulnerable.
My question is that since it is such a vulnerable thing and there's a lack of diversity, is there a way to create diversity so that we have a wider variety of species and things are more likely to survive?
Well, I know that the banana flavor that we have in banana candy is the flavor of the banana that no longer exists. That's why banana candies don't taste like bananas.
Now that's interesting.
Because that changed around World War II. Now that I'm not a faint echo in the background. Allan, pay attention to the buttons on the table.
Well, I think World War II was actually similar to where they got a similar situation happened with the seed vault. If it was World War II, World War I, I can't remember exactly right now. There were a group of scientists that had a bunch of the seeds, and they ended up having to barricade themselves in because there was no food anywhere else, and people were trying to break in to get the seeds to eat. And they said, no, after everything, we can plant these and there'll be more food. We just have to wait a little bit.
Well, it's welcome to preserving future ideas. And Max, you are dead on the money. There are a number of companies, happily, that are looking at producing and comparing and possibly even hybridizing a wide variety of the other varieties that are available, in the contingency that one virus that, we're not going to pick on humans for a chance. We're going to pick on a plant virus. One virus gets loose, and you have like the Irish potato famine. You have a really heavy dependency on that one crop, and you've planted a lot of it because the way we produce food isn't very natural.
No.
If you go out to a field, you'll notice there's just all different kinds of plants all mixed in there together. And that's kind of how the natural world works. You don't really wind up with a big wheat field. It's a wheat field with all kinds of other stuff growing in there with it. That's what we're looking at. So Some things are better in the cold. Some are better in the hot. Some like it wet. Some like it dry. You can have probably, in certain plants, you can have 10, 20, 30, 50, 100 different varieties scattered around the world. I love to pick on tomatoes. Huge variety in tomatoes. Huge variety. There's different colors. There's heirlooms. There's all kinds of new varieties, old varieties. And a lot of places around the world, once tomato was introduced to them, kind of came up with their own varieties. We are constantly playing with chili peppers. Another one that is constantly being bred and crossbred and new varieties and local favorites. You get into onions and garlics and all of these plants. And guys, we're all still talking about genes. Now, if we elevate this above the seed bank just a little bit, because you're right, Max, that we are trying to protect that diversity. Because what if, we got to throw in the horrible what if, we do have a really terrible hurricane season and the citrus fruits are damaged. And then it's followed by a terrible, as we just got through, as you picking on the ice here, we just had a massive ice storm. Okay. We just lost those trees.
Yeah.
Well, are they gone or are there hardier varieties? Are there other types that could take the cold better?
This makes me think of something rather intriguing. Rather than just also focusing on protecting what is, we also have the technology, as we alluded earlier before, like way back several shows ago, we talk about resurrecting what was.
Exactly. So here we go. Let's pick on an orange tree for a moment. And for the sake of CRISPR, let's say we could add a little Satsuma genetic material, because a Satsuma is basically a mandarin orange that can handle slightly colder temperatures. Satsumas grow as far north as Louisiana. Now, you're not going to find them in Tennessee, but it's a little hardier. So we could produce a navel orange that wasn't quite so vulnerable. Or we could wander back into time. And of course, we're looking at things that were directly impacted by humans. We know that mastodons and mammoths and that kind of thing, the woolly rhino, were all part of the last ice age. Well, a lot of that territory, you get into northern Alaska, go to Siberia, you have a lot of that same environment. But they're now lacking these linchpin creatures. So they're more locked in snow, more locked in ice. Because let's face it, a woolly mammoth was the herbivore version of a bulldozer. 4 inches of snow, it giggled. 4 feet of snow, it was still plowing along just fine. Now, we still have some megafauna like a muskox. But what if, and there is actually a company that has now filed to use the CRISPR technology to basically take the genome of an Indian elephant, and we're going to play Mr. Dino DNA
Dino DNA.
And we're going to add the mastodon or the mammoth features that we've been able to glean from the frozen samples. And the end result should be a mammoth-like, very much a mammoth-like creature that would be a big old woolly rhinoceros or elephant or whatever species we opted for. This would be an interesting way of preserving things like Galapagos tortoise species variations. How about bringing something back like the dodo? Well, the dodo we pick on the dodo as being a big dumb bird, but he really wasn't a big dumb bird. It was actually a relative of the pigeon and was indeed a really big bird, and it just never seen humans before. And when we discovered islands, people discovered that the animals didn't run in, absolute fear of you because they had no idea what humans were or what they were capable of, and the fact that we'd probably eat them.
Yeah.
And, it's kind of been our history. So now one of the problems they're running into in the islands where dodos were known to be is the dodo was such a large bird, it could handle eating the nuts from the trees around there. And they had kind of a relationship where the dodo ate the nuts, digested the nuts, and now the nuts were able to sprout. So the trees could keep renewing themselves.
Well, that brings up an interesting point of if we bring these animals back, what are the ecological implications of that? Do we have the proper place for them to live? Because if we don't have many records of them, we don't know what impact they'll have on an area or what area the impact would have on them.
Well, what about the thylacine? It didn't go extinct until 1910.
Because
so we knew
people-
exactly what was going on then. So you're now missing your apex predator from Lower New Guinea, Australia, and Tasmania. So we knew they were there. We're the ones that hunted them out. And they have specimens preserved in alcohol that preserves the DNA. So in theory, we could bring back something. Kind of an interesting point of view, and really easy to make a leap right over to Jurassic Park, isn't it? But in reality, that's this is very long and arduous science. We're not imminent, but is this something that is going to happen? And I look directly at Max and go, looking at what the genome progresses and does, this is going to be a dramatic topic through the entire length of your life.
Dino DNA to be determined.
Well, ladies and gentlemen, it was on the news. We've enjoyed talking about a little gene manipulation and CRISPR with you. And we will talk to y'all next time.